Imagine for a second that you've been relying on the exact same maps of the world for centuries. I mean, you know every single continent, every mountain range, every tiny little island.
Right. The borders are totally settled.
Exactly. But then, one day you zoom in with a brand new satellite and you realize you completely missed an entire interconnected ocean spanning the globe.
Oh, wow.
Yeah. And it was there the whole time, right? Flowing right between the land masses. But your old maps just, they just weren't equipped to show it.
It sounds totally like science fiction, I know. But that is precisely the scale of what we are looking at today. Only, well, we aren't looking at the Earth. We are exploring the geography inside you.
Yeah. And this whole paradigm shift, it started with this landmark 2018 paper published in Scientific Reports by Benias and his team. Basically, they realized the medical textbooks have been giving us an incomplete picture of human anatomy for a century.
Your literal century, yeah.
So the mission of this deep dive today is to explore that newly recognized, massive, fluid-filled anatomical space. It's called the interstitium.
And, for context on why this is just so groundbreaking, we have been cutting up human tissue and looking at it under microscopes for hundreds of years.
Right.
So the sheer audacity of the idea that we could have missed a massive body-wide fluid structure hiding right beneath our skin and in our gut is staggering.
Which brings up the obvious question for you listening. How on earth do you miss an ocean inside the human body?
Well, the answer really comes down to a fundamental change in the tools we use to look at ourselves. The researchers, they didn't find this with a standard scalpel or, a traditional light microscope. They used a technology called PCLE.
Wait, PCLE, what exactly does that stand for?
It stands for probe-based confocal laser endomicroscopy. It's a mouthful, I know.
Yeah, just a bit.
But the brilliance of PCLE is that it lets us look at living tissue in its natural state. Because normally if a doctor wants to see your cells, they have to physically remove a piece of you.
Right, biopsy.
Exactly. But PCLE allows for real-time in vivo histologic imaging at a depth of about 60 to 70 micrometers down.
So it's essentially like taking a microscopic submarine, injecting it into your tissues, and watching it explore live while your blood is still pumping.
That's a perfect analogy, yeah. During a regular endoscopy, like when a doctor is examining the digestive tract, they use this probe to look deep into the tissue wall.
But I'm guessing human tissue doesn't just naturally glow under a camera, right?
No, it definitely doesn't. So to make the microscopic structures visible, the medical team injects a fluorescent dye. It's called fluorescein. They inject it directly into the patient's bloodstream.
Okay, got it.
Then they use a highly targeted laser from the tip of the probe to illuminate the tissue. The laser hits the fluorescein dye and causes it to light up brilliantly.
And so the aha moment of discovery happened when researchers were looking at a patient's extrahepatic bile duct.
Yes, the tube that carries bile away from the liver.
Now, the textbooks dictated that they should see a solid, dense wall of collagen tissue there. But when the laser hit the dye, they didn't see a solid wall at all.
Not even close. It revealed this bizarre alien reticular pattern, like a net-like maze.
A maze?
Yeah, there were these dark, branching bands of tissue, completely surrounded by bright, glowing spaces filled with the fluorescein dye. And these glowing fluid pools were roughly 20 micrometers wide.
Wow.
The researchers immediately recognized that this just didn't match any known anatomical structure. I mean, it wasn't a network of capillaries, and it wasn't the tiny lymph vessels we call lymphangials either.
It was an entirely undocumented landscape.
Exactly.
So if you're listening to this and wondering, why your doctor never mentioned this massive fluid network before, the answer involves a really fascinating twist of medical history.
It really does.
Because the very methods pathologists used to study tissue for the last 100 years were actually, they were destroying the evidence of this space before anyone could see it.
Right. It comes down to the mechanics of traditional histology. When a surgeon takes a biopsy, the tissue begins to degrade immediately. So to stop that decay, the lab treats the slice of tissue with harsh chemical fixatives, usually formaldehyde. And then they use alcohol to pull the water out so the tissue can be embedded in paraffin wax and sliced, paper thin.
But that vital dehydration process literally sucks all the fluid out of the sample.
Exactly. It's an artifact of fixation.
I like to think of a wet kitchen sponge. I mean, it's full of open, fluid-filled pockets. But if you take that wet sponge, put it in a vacuum-sealed bag, and suck out absolutely all the water and air, the internal structure collapses.
Right. It turns into a dense, solid brick.
Exactly. And for generations, pathologists were looking at these vacuum-sealed sponges under their microscopes. Because the chemical preparation collapsed all those fluid pockets, the tissue just looked like a thick, solid wall of collagen.
And they logically assumed that's just how the body was built.
So how did they finally prove that this solid wall was just, a man-made illusion?
Well, they had to find a way to capture the tissue while the fluid was still inside it. They needed to freeze it while it was still alive, essentially, before the fluid had a chance to drain away.
And they accomplished this during Whipple surgeries, right?
Yeah, Whipple surgeries are highly complex procedures often used for pancreatic cancer. While the patient was on the operating table with blood still flowing, the researchers took samples and immediately hit them with an aerosol-based freezing spray.
Just instantly freezing it.
Yes, in milliseconds. By dropping the temperature that fast, the fluid inside the tissue forms microscopic ice crystals instantly. It locks the physical architecture in place.
Oh, that's brilliant.
Then they took that frozen tissue into a specialized cold room and carefully cut serial sections, slicing it every 5 micrometers deep, down to that 60 to 70 micrometer depth. And the original structure was perfectly preserved.
So the bright pools of fluorescein were still trapped exactly where the live imaging showed them.
Exactly. Nestled inside that dark branching network.
Let's do a quick recap of this first part, because this is wild. So live PCLE imaging revealed a mysterious fluid-filled pattern. Then rapid freezing preserved that live anatomy. And that made us realize standard biopsy techniques were actually collapsing a massive fluid space into what looked like dense collagen walls.
That's a perfect summary.
So here's a question for you listening. If our standard preparation methods blinded us to a massive bodily structure for a whole century, what other invisible systems might be hiding in plain sight, just waiting for the right lens to view them?
It really makes you wonder.
It does. But, okay, once they proved this fluid-filled network was real, they had to figure out what it was actually made of. Right? We're moving from the how to the what.
Right. And the microanatomy of the space is wildly complex. It's not just a random void where fluid happens to pool. It has a highly engineered architectural blueprint.
Like a structural lattice.
Yes, a meticulously woven biological lattice. Remember those dark branching bands they saw on the initial PCLE imaging?
Yeah, the dark maze parts.
Yeah. Well, those are actually thick bundles of fibrillar collagen. They verified this using a technique called second harmonics generation imaging.
Oh, what is that?
It's a specialized type of microscopy where 2 photons of laser light interact with highly organized, dense fibers like collagen, and it causes them to emit a very specific dark blue glow.
Okay, so collagen is basically the steel rebar of the body. It provides the structural strength to keep our tissues from tearing.
Exactly. But if our tissues were only made of rigid steel rebar, we wouldn't be able to bend our joints or expand our lungs.
Right, we need to stretch.
Which is why, right alongside that rigid collagen, they found elastin fibers. They confirmed the elastin because it has a unique property called autofluorescence.
Meaning it glows on its own.
Mm-hmm. It naturally emits a cyan color under certain light without needing any dye. Plus, it stains a deep black when treated with an elastic Van Giesen stain. So the environment is this beautiful synergy. The collagen acts as the strong, rigid scaffolding, and the elastin provides the stretch and recoil, like a bungee cord.
I love that bungee cord analogy. But scaffolding in the human body is rarely left alone. There are cells living inside this lattice.
Oh, absolutely. And to see them, they had to zoom way, way down to 80 nanometers using transmission electron microscopy.
That is unbelievably tiny.
It is. And what they found were these incredibly thin, flat, spindle-shaped cells clinging to the collagen bundles.
But they weren't standard blood vessel cells, right?
No, and that's key. Defining what these cells actually are requires ruling out what they aren't. Normally, the internal fluid channels of your body, like your blood vessels, are lined with endothelial cells.
Okay.
Those vessel cells have very specific microscopic machinery. For instance, they contain pinocytotic vesicles.
Sure, what exactly?
Basically, tiny cellular bubbles used to actively transport fluids back and forth across the cell wall. Endothelial cells also have Weibel Palade bodies, which are essentially storage units for specific proteins. And crucially, they always sit on a foundation called a basement membrane.
But cells in the interstitium didn't have any of that.
None of it. No fluid transporting bubbles, no storage units, no basement membrane.
So if they aren't standard blood vessel cells, what's their function?
To figure that out, the researchers looked at the proteins on the outside of the cells, like their chemical ID badges. Normal blood vessel cells carry an ID badge called CD31. These cells tested completely negative for that.
Interesting.
Instead, they were uniformly positive for a marker called CD34 and another structural protein called vimentin.
And what does CD34 tell us?
In the biological world, carrying the CD34 marker suggests that these are a completely novel type of fibroblast or possibly even a mesenchymal stem cell. They seem to be totally unique to this specific interstitial space.
And the way these cells arrange themselves leads to one of the most bizarre findings in the entire paper. These unique CD34 cells only line one side of the collagen bundles.
Yeah, that asymmetrical lining breaks a lot of conventional biological rules.
How so?
Well, because the cells only cover one side, the unlined side of the collagen bundle leaves its bare matrix proteins directly exposed to the fluid flowing through the space.
And for those of us who don't, spend our weekends reading cellular biology textbooks, having bodily fluid flowing directly over naked protein without a protective cell barrier is exceedingly rare, isn't it?
It almost never happens. Prior to this, scientists only really observed it in very specialized environments, like the kidney's glomerulus, where blood is aggressively filtered, or in the liver space of DISI.
So why does it matter that the protein is exposed?
Because collagen is a highly charged molecule. When you have biological fluid flowing directly over a bare electrically charged surface, it creates a highly reactive environment. It is not just an inert pipe carrying water. That charged surface likely interacts with the proteins and cells floating past it.
Let me jump in here with a quick recap of this section. So the interstitium is a lattice of strong collagen and stretchy elastin, and it's lined asymmetrically by unique CD34 positive cells, which creates this exposed electrically charged highway.
Perfectly said.
So think about this for a second. Imagine fluid flowing directly over the bare charged building blocks in your body. How do you think immune cells traveling this route react to interacting with raw collagen?
It's a huge question.
Okay, we are about halfway through, so let's do a fast 5-minute bullet recap to make sure we're all tracking. Number 1, live imaging uncovered a massive hidden space. Number 2, freezing live tissue proved that standard biopsies were chemically collapsing it. Number 3, we found it's a collagen and elastin lattice. And number 4, it has exposed protein surfaces and strange CD34 lining cells.
That covers the structural anatomy perfectly.
But I'm going to push back a little here. I get that it's a fascinating structural slip and slide. But does this microscopic shock absorber actually do anything for our survival? Or is it just like biological bubble wrap for our organs?
Viewing it as just biological bubble wrap drastically underestimates its role. It does act as a shock absorber, certainly. The researchers mapped its distribution, and it exists everywhere in the body subject to rhythmic or intermittent compression. It's woven throughout the dermis of your skin. It forms the submucosa of your entire gastrointestinal tract, which is constantly squeezing during digestion. It's in the urinary bladder, around the lungs, and it coats the major fascial planes of our muscles.
Basically, any tissue that expands, contracts, or handles physical stress relies on this fluid-filled sponge layer.
Right. But the physical cushioning is just a byproduct of its true function. It is fundamentally tied to our internal fluid dynamics. The researchers actually proposed that this space is a pre-lymphatic conduit.
Pre-lymphatic meaning? This space is the primary source of lymph. It's where interstitial fluid gathers before becoming actual lymph in the main vessels.
Exactly. It's the tributary that feeds the main river of our immune system. And they verified this connection through a brilliant piece of medical detective work involving a colorectal tattoo.
Oh, I love a good medical detective story. Let's hear it.
So before certain colon surgeries, doctors will inject a black tattoo pigment into the submucosa of the patient's colon to visually mark the exact site for the surgeon to find later.
Just a standard tracking procedure.
Right. But the fascinating part happened after the surgery. When pathologists examined the resected tissue under a microscope, they found macrophages, which are essentially the immune system's cellular pac-men that roam around eating foreign debris, filled to the brim with this black tattoo pigment.
Let me guess. Those macrophages were hanging out inside this exact interstitial space.
Yes, but the crucial piece of evidence is where those macrophages went next. The researchers found those exact same pigment-filled immune cells in the patient's distant mesenteric lymph nodes.
Oh, wow. So the macrophages ate the ink, hopped into the interstitial fluid channel, and rode the current all the way to the lymph nodes.
Exactly. It definitively proves the interstitium is a direct functional transit network for the immune system.
That is incredible.
And taking that mechanical understanding a step further, this transit network finally explains a massive medical mystery known as third spacing.
Oh, right. Anyone who has dealt with severe swelling or edema has probably experienced this.
Yeah. When a patient goes into liver, renal, or heart failure, or gets post-operative lymphedema, their tissues swell with massive amounts of fluid. Doctors refer to this as fluid moving into the third space. But for a long time, the exact anatomical boundaries of that space were, well, somewhat abstract.
But now we can actually see it. The researchers provided visual evidence.
They did. They examined resected bowels from incarcerated hernias, where the bowel is totally physically obstructed and fluid drainage fails. Under the microscope, they saw the normally compact collagen bundles splayed wide open.
Like balloons.
Yes, filled with a pink proteinaceous fluid. The interstitium literally acts as a massive overflow reservoir when fluid dynamics fail.
Okay, quick recap time again. So the interstitium isn't just structural. It's a dynamic pre-lymphatic river. It shuttles immune cells, as proven by the tattoo pigment, and it's the physical location where fluid builds up during edema and organ failure.
Right.
So the next time you get a severe blister or twist your ankle and see it swell up, picture this exact microscopic sponge expanding with fluid. Does visualizing this structural mechanism change how you view your body's healing process? It definitely does for me.
It's a powerful visual.
But unfortunately, a transit network built to shuttle immune cells and handle fluid overflow can also transport much more dangerous passengers.
Right.
Which brings us to the dark side of this discovery.
Yes. The cancer implications outlined in this paper represent a major shift in oncological theory. If this space is a fluid superhighway leading directly to the lymph nodes, it provides a terrifyingly efficient route for tumor cells to spread.
Because what stops a tumor cell from just jumping on the on-ramp?
Nothing, really. And this mechanism brings clarity to a clinical observation about tumor staging that has frustrated oncologists for decades.
We're talking about T1 versus T2 tumors here.
Exactly. Based on the source material, a stage T1 tumor sits locally on the surface. It's like a weed growing on the top of the sidewalk, for example, on the very inner lining of the stomach.
But a stage T2 tumor is invasive. It breaks through that top layer, and its roots penetrate down into the submucosa in the stomach, or the deep dermis in the skin.
And doctors have always known that the moment a tumor becomes T2 and hits that deeper connective layer, the risk of metastasis, the cancer spreading to the lymph nodes, massively skyrockets.
But the confusing part was they couldn't always explain how. Because sometimes pathologists couldn't find any cancer cells actively invading the local blood vessels or lymph vessels.
Right, and the paper details two specific case studies that highlight this mystery perfectly. One involved a poorly differentiated gastric carcinoma, a really aggressive stomach cancer, and the other was a malignant melanoma on a patient's arm.
And both of these were stage T2. They had successfully invaded the submucosa and the dermis layers.
Yes. But when the medical team examined the tissue, neither cancer showed any evidence inside standard lymph or blood vessels. No classic lymphovascular invasion at all.
Yet both had successfully metastasized to the lymph nodes anyway.
Exactly. The missing link was how they traveled there. And the specialized microscopy revealed the chilling answer.
What did they see?
Under the microscope, they saw individual cancer cells and tiny clusters of them, basically infiltrating the fluid-filled interstitial space, isolating the collagen bundles.
Just floating freely in the fluid highway?
Yes. And the transit mechanism relies on the host's own bodily functions, because this space handles rhythmic compression, right? So the normal peristalsis of the stomach digesting or the physical flexing of the arm muscle beneath the melanoma literally pumps the fluid.
Oh, wow. So every time the muscle flexes, it physically pumps the interstitial fluid and the floating cancer cells down the channel directly to the lymph nodes.
Exactly. The physical compression drives the metastasis.
That is a dark realization. Our own physical movements pump the cancer through the system, but mechanically, it explains the aggressive nature of T2 tumors flawlessly.
It really does. And the implications for pathology extend beyond cancer, too, into the realm of chronic scarring.
Oh, right. Let's revisit those mysterious CD34 cells from earlier, the ones lining the collagen bundles.
Right. The researchers note that if these unique CD34 cells are precursors to myofibroblasts, they are basically the body's emergency construction workers.
They lay down dense collagen to create scar tissue.
Exactly. So severe fibrotic diseases, where the body uncontrollable creates scar tissue, might actually be diseases of the interstitium.
The paper specifically points to conditions like keloids, right? Those overgrown, raised scars you get on skin under high tension. Or deeper issues like scleroderma or inflammatory bowel disease.
Yes. In all these cases, continuous mechanical pressure drives these CD34 cells to overproduce collagen, essentially clogging up the fluid channels with concrete.
Time for another quick recap. The fluid-filled nature of the interstitium explains why cancers become so deadly once they reach the submucosa or dermis. They gain access to a slip and slide direct to the lymph nodes. Plus, its unique lining cells might be the originators of severe fibrotic scarring diseases.
Spot on.
So a question for you listening. If the physical compression of our organs helps pump cancer cells down this highway, could targeted changes in tissue tension or fluid dynamics one day become a form of cancer treatment?
That is the ultimate goal. Reevaluating the submucosa, the dermis, and our fascia as active, fluid-filled channels instead of dense walls changes how we interpret every bodily function.
I mean, think about the peristalsis of your gut. We're taught it's just moving food from point A to point B. But if the whole gut wall is filled with this flowing fluid, that physical wave might be a massive communication network, sending hormonal and immunologic signals proximally to distally through this internal ocean.
And the researchers leave us with a final, forward-looking concept derived directly from their conclusion.
Let's hear it.
If this interstitial fluid flows around the whole body, constantly picking up cellular signaling, pro-fibrogenic molecules, and even stray tumor cells long before they reach the general bloodstream, what if we could sample it?
Oh, wow. Instead of drawing blood.
Imagine a future medical checkup where, instead of a blood draw, a microscopic needle samples your interstitial fluid. It could detect the very earliest molecular whispers of cancer or inflammatory disease, catching bad actors before they even reach the lymph nodes.
That is just mind-blowing. The promise of an entirely new diagnostic frontier hiding right under our skin. It brings us right back to those old maps we started with. We simply didn't know the ocean was there, but now that we have the right satellite, we can finally navigate it. Thank you so much for joining us on this deep dive into the hidden spaces of the human body. It's a powerful reminder to never stop questioning the established maps. Keep exploring, and we'll catch you on the next deep dive.